DNA stands for deoxyribonucleic acid, a molecule found in cells that carries the genetic instructions necessary for the growth, development, functioning, and reproduction of all known living organisms and many viruses. It consists of two long chains made of nucleotides, each containing a sugar phosphate backbone with nitrogenous bases adenine, thymine, cytosine, and guanine attached. These bases pair up in a specific manner, A with T, and C with G, across the two strands, forming the famous double helix structure discovered by Watson and Crick in 1953. DNA holds the genetic information that is passed from parents to offspring and is responsible for the diversity and characteristics of living organisms. RNA, or ribonucleic acid, is another essential molecule involved in various cellular processes. Similar to DNA, RNA is composed of nucleotides, but it typically exists as a single strand. RNA serves as a messenger, carrying instructions from DNA for controlling the synthesis of proteins, as well as playing roles in other cellular functions. There are different types of RNA, including 1. Messenger RNA, mRNA. It carries genetic information from the DNA in the nucleus of a cell to the ribosomes in the cytoplasm, where proteins are synthesized based on this information. 2. Transfer RNA Turner. It assists in protein synthesis by bringing specific amino acids to the ribosome, where they are linked together to form a protein chain. 3. Ribosomal RNA, rRNA. It is a major component of ribosomes, the cellular machinery where proteins are assembled. Apart from these, there are other specialized forms of RNA involved in various regulatory functions within cells, such as microRNA, mRNA, and small interfering RNA, siRNA, which play roles in gene regulation and silencing. RNA plays a crucial role in the expression of genetic information and the regulation of various cellular processes, working closely with DNA to ensure proper functioning and development of organisms. A gene is a basic unit of heredity that carries information determining various traits, such as physical characteristics or predisposition to certain diseases, by providing instructions for building proteins. Genes are made up of DNA sequences and are passed down from parents to offspring during reproduction. They play a fundamental role in the functioning and development of living organisms. A mutation is a permanent alteration in the DNA sequence that makes up a gene. It can occur due to various factors such as errors during DNA replication, exposure to radiation, chemicals, or environmental factors. Mutations can take different forms, including substitutions, insertions, deletions, duplications, frameshifts, inversions, or translocations. Mutations can have diverse effects. Some mutations are harmless, causing no noticeable changes in an organism, while others can have significant impacts. Beneficial mutations may contribute to genetic diversity and occasionally lead to advantageous traits. However, harmful mutations can result in genetic disorders or diseases. Understanding mutations is crucial in fields like genetics and medicine, as it helps in studying diseases, tracing evolutionary relationships, and developing treatments or interventions for genetic conditions. There are several types of mutations that can occur in DNA. 1. Substitution. One base pair is replaced by another. 2. Insertion. Extra base pairs are inserted into a DNA sequence. 3. Deletion. Base pairs are removed from a DNA sequence. 4. Duplication. A section of DNA is duplicated, leading to extra copies of that segment. 5. Frameshift. Insertion or deletion of base pairs that alters the reading frame, changing the way the sequence is interpreted. 6. Inversion. Reversal of the orientation of a segment of DNA. 7. Translocation. Movement of a segment of DNA from one chromosome to another. These mutations can occur spontaneously or be induced by external factors like radiation, chemicals, or errors during DNA replication or repair. They can have various effects on an organism, ranging from negligible to significant alterations in traits or functions. Gene-therapy is a medical approach aimed at treating genetic disorders by modifying or introducing genetic material into a person's cells. Disorders treated. Inherited disorders. cystic fibrosis, Duchenne muscular dystrophy, Huntington's disease, cyclic cell disease, thalassemia, Leber congenital #amaurosis and retinitis pigmentosa and hemophilia are being targeted. Cancer. A treatment option for certain types of cancer. It may involve modifying a patient's own immune cells to better recognize and attack cancer cells, immunotherapy, or targeting specific genes involved in tumor growth. Neurological disorders. Conditions such as Parkinson's disease, Alzheimer's disease, amyotrophic lateral sclerosis (ALS), and certain types of epilepsy to mitigate symptoms or halt disease progression. Metabolic disorders, familial hypercholesterolemia and metabolic storage diseases. Methods of gene therapy. Viral vectors, modified to be harmless adeno-associated viruses (AAV), and lentiviruses are commonly used viral vectors carriers. Non-viral vectors, non-viral approaches like liposomes, nanoparticles, or other chemical carriers to introduce genetic material into cells. They are considered safer but often less efficient compared. CRISPR-Cas9 and genome editing. This technique involves precise editing of DNA sequences to correct genetic mutations causing diseases. CRISPR-Cas9 ACTS as molecular scissors, to cut and modify DNA at specific locations. Oligonucleotide therapies. These therapies use short sequences of nucleic acids, oligonucleotides, to modify gene expression, correct RNA function, or regulate protein production. Cell-based therapies. Modified cells, such as immune cells or stem cells, are introduced into the body to replace or repair damaged tissues. Procedure for gene therapy. Identification of target gene. Delivery vehicle selection. Choosing the appropriate method to deliver the therapeutic gene into the patient's cells. This could involve viral vectors, non-viral methods, or genome editing tools. Modification of genetic material. The therapeutic gene or gene editing tools are prepared for delivery. For instance, if using a viral vector, the therapeutic gene is inserted into the vector. Administration. The modified genetic material is introduced into the patient's body. This can be done through direct injection into specific tissues or intravenous infusion. Integration or expression. The delivered gene is incorporated into the patient's cells and begins to express the desired protein or regulate gene expression, aiming to correct the genetic defect or provide therapeutic benefits. Monitoring and follow-up. Patients are monitored to assess the therapy's effectiveness, potential side effects, and long-term outcomes. Complications and challenges. Immune response. The body might react to the introduced genetic material or delivery system, leading to immune reactions that could reduce the therapy's effectiveness or cause adverse effects. Off-target effects. Genome editing techniques like CRISPR-Cas9 may inadvertently modify genes other than the intended target, potentially causing unforeseen consequences or side effects. Limited duration of effectiveness. In some cases, the therapeutic effects of gene therapy might not be long-lasting, requiring repeated treatments or booster doses to maintain benefits. Ethical concerns. Gene editing raises ethical questions, especially concerning germline editing, modifying genes in sperm, eggs, or embryos due to concerns about hereditary implications and the potential for unintended consequences in future generations. Risk of insertional mutagenesis. When using viral vectors, there's a risk of unintended insertion of the therapeutic gene into the wrong place in the genome, which could disrupt normal gene function and potentially lead to cancer or other complications.